There are many different types of molecules that make ECM. The first category we are going to talk about are the glycosamine glycans, also referred to as GAGs. Let's look at some of the features of these molecules. I would also like to point out that there are two main classes of ECM. GAGs make, for example, polysaccharides, including glycans. They make one category, and also proteins, for example, collagen, elastin, fibronectin, and laminin. That's a second category of ECMs. We are going to first of all talk about GAG molecules. GAGs are proteoglycans. These molecules can form highly hydrated gel-like substance, in which fibrous proteins are embedded. Polysaccharide gels resist compressive forces. We have talked about compressive forces. The ones which are, for example, pressing against your skin are the compressive forces. And also, these gels permit diffusion of nutrients, metabolites, and hormones between blood and cell, whereas collagen fibers, or the protein part of the ECM, strengthens and organizes the matrix into rubber-like or elastic-like fibers or sheets, which gives the matrix resilience and keeps the cells in appropriate locations. And also, these type of molecules resist basically tensile forces. Here is a diagram of two different types of ECMs are shown in this diagram. Here, for example, this is basal lamina. Basal lamina, we'll talk about it in more detail. This ECM basically is responsible for attachment of epithelial cells to it. Below basal lamina, there's connective tissue. It is full of different type of carbohydrate ECM molecules. For example, hyaluronan, proteoglycans, and other glycoproteins. Also, this contains different elastic fibers, which are shown here in green. In this part of this tissue, we also have macrophages. We talked about them when we were discussing immune system. Here you can see there's a capillary and also the endothelial cells which form the lining of a capillary through which blood flows. Also, you would notice that there is a dark object here. That's the mast cell, and these dark granules are basically granules that contain histamine, which is released in case this tissue is injured, which is responsible for bringing in other cells of the immune system. And they're also different, although you cannot see the structure. This is basically the hydrated gel in which these cells and other molecules are suspended. Glycosamine glycans, or GAGs, these are unbranched polysaccharide chains of repeating disaccharide units. One of the two sugars in the repeating disaccharide is always an amino sugar, which basically means that there's an amino group attached to one of the sugars. Either it is N-acetyl glucosamine or N-acetyl galactosamine. Here in this diagram, the red part here is basically N-acetyl galactosamine, and it is attached to a sulfate group. We will talk about the significance of sulfate attachment to these molecules later on. But also, I would like to mention that the second sugar, in most cases, is either a uronic acid or glucuronic acid. These molecules are the ones which are in most cases, in many cases, they are sulfated. So that gives these molecules negative charge. GAGs are highly negative because of these sulfate groups that are attached to them. Because of that, they can attract positively charged ions, for example, sodium ions, and result in absorption of water and thereby forming these gels. There are four main groups of GAGs that are distinguished according to the sugars, the type of linkage between the sugars, number and location of the sulfate groups. The first category is hyaluronan. The second category is chondroitin sulfate and dermatan sulfate. The third category is heparan sulfate. And the fourth category is keratin sulfate. We look at these molecules individually, but in a later module. But now I would like to mention that polysaccharide chains are too stiff to fold into compact globular structures like polypeptide chains. They form these hydrophilic gel-like structures because gags adapt a highly extended conformation, and they occupy huge volume relative to their mass to form these gels. And they can form these gels even at a very low concentration. The high density of negative charges, as I mentioned, can attract a cloud of osmotically active positively charged ions, for example, sodium ions, causing large amount of waters to be sucked into the matrix. This creates a swelling pressure or turgor pressure that can enable matrix to withstand compressive forces. For example, cartilage matrix in the knee joint can support up to a pressure of hundreds of atmospheres. This is how strong they are in resisting the compressive forces. In connective tissue, gags constitute less than 10% of weight, the dry weight of the fibrous proteins, since they can attract a lot of water. Porous gag, hydrated gels, the chains that fill up the extracellular space, they can provide the mechanical support, for example, resisting compressive forces. Dermatan sulfate disaccharide gene, if it is mutated, it causes short stature, prematurely aged appearance, and generalized skin defects and joint and muscle defects. Here you can see there is in this photograph, you can see a comparison of different molecules. Here, for example, we have a globular protein which can fold tightly into a globular structure. A molecular weight here of this protein, I should mention that these are basically drawn to scale. So it's not that some are larger and some are smaller. These proteins, these molecules are drawn relatively to the scale. Molecular weight is 50,000. Here we have glycogen molecule. Glycogen we have talked about, it's also carbohydrate. Here we have a spectrin molecule. It's part of the cytoskeleton. Molecular weight is 460. As compared to collagen, and collagen drawn here has a molecular weight of 290. This is a collagen molecule. Here we have hyaluronan, which is type of gag. Its weight, molecular weight is 8 into 10 raised power 6. And look at the amount of volume it is taking. Volume is, of course, a three-dimensional quantity. When we are measuring something in 3 dimensions, this hyaluronan can basically occupy large volume compared to its proteins. So these gags absorb water. They can form hydrating gels and occupy large volumes as compared to their weight.